Good morning, everybody. Thank you for joining us today, I think, for our first DNA and our first NMR talk of this series. So I'm super excited that we're going to be hearing from Dr. Cameron Mackereth today. He started his career in Canada, in, I believe, Vancouver, for his PhD, where he started thinking about protein families and NMR. And then he continued at EMBL in Heidelberg in Germany for his postdoctoral work, where he did wonderful NMR work on kind of splicing mechanisms and other complexes. And now is in France at the University of Bordeaux, where he works on wonderful new NMR technologies and has done really interesting work on nucleic acids. So today we're going to be hearing a bit about his work on DNA NMR structures. And one of these was actually a CASP target. So I think it's D1273. Weirdly, I know the number was the CASP target, our only DNA target, but hopefully we'll have more in the future. All right, take it away.
All right. Well, thank you very much. It's really a fun opportunity to talk a little bit about the experimental structural aspects of looking at DNA and really, hopefully, by the end of this, you'll see what we can see by NMR structures and other aspects, but also how diverse probably DNA structure will end up being, and not just double-stranded. So hopefully you get that from today's talk. So this is a picture of Bordeaux, unfortunately not the view from the university. Um, Before I start anything, I would just like to actually start with the acknowledgements. So today I'm going to be talking a lot about the structure determination, and I did a lot of NMR work with the help of various people. But you'll see during this talk, most of my slides have a black background, but when there's a white background, keep note. And if you have a particular question, I will probably ask Eric, who will give some advice because he did a lot of the MD and structural analysis from my group. We also had some DNA synthesis that was important. That was thanks to Brune. And it was a collaboration with people in other places in Canada, actually, as it turns out, one at York University in Toronto, so the lab of Philip Johnson, and in particular his PhD students, Emily and Rahith, and the University of Sherbrooke, and Philippe and Mindat. So I'll start right into what we had starting this project. And what we noticed is that there's a lot of very nice aptamer structures. So aptamers, I'm not really going to talk too much about that today, but it's a way to select DNA sequences or RNA sequences, for example, but I'm going to stick to DNA sequences that are specific for a target and with high affinity and various other properties depending on how you decide to do the initial screen. And there are many very good and functional aptamers, but usually, and I'll use this pointer here, usually the extent to which the structural details are shown are usually along the lines of an M-fold type prediction. So you see often regions of double strand, and you see some bulges or loops, stem loop structures. And that's usually as far as it goes because, to be honest, the real point of a DNA aptamer is that it's functional and tells you maybe by binding to a particular target for a diagnostic or therapeutical reason. So it's more about the function and not necessarily how it does it. But we noticed that actually you could probably gain a lot more insight by knowing how it interacts, because this will give you the possibility to change what it's binding to, or really just optimize it in different ways for the given purpose that you'll be using it for. So from the paper back in 2018, we looked at these potential structures. I'll be talking today about dopamine, so the very first one on the left from figure 1 in this paper. And it was used to make this very nice sensor in this article. And we noticed here the structure, as I said before, is usually a stem loop type with bulges. And somehow it was expected that it would bind specifically to dopamine. So this was the starting point. We knew that the DNA aptamer was functional, and we knew that it bound to dopamine, and that was about it. So, so we decided to start from here and see what it looked like by NMR spectroscopy. And one of the fairly rapid things you can do when looking at a DNA structure by NMR is to see in solution what type of structure is happening. For example, is it structured or not? And the easiest way to do that is to look at these hydrogens that are attached to nitrogen on the DNA bases. And we look at those in particular because in a base that is free in solution, so if you just had a guanine base, this hydrogen would actually rapidly exchange between bound to the base and exchanging with the solvent, which is usually water. So because of that, they're actually not usually visible in an NMR spectrum. So the signal gets averaged or it just isn't there for long enough to be detected by the NMR experiment. So when we do see this particular hydrogen, we know that it's likely in a hydrogen bond type scenario because then it's actually stabilized. And when it's in a hydrogen bond, it doesn't exchange as rapidly and it behaves much more like the hydrogens that are attached to carbon, for example, on the base. So we can actually see these hydrogens. And in a more practical sense, what this means is that, for example, we often look at DNA aptamers that were, because of the selection process, they are unfolded, primarily unfolded, in the absence of ligand. And when you add a ligand, you see the appearance of all of these specific peaks, we call them, so these NMR signals. And this particular region of the hydrogen, or proton, we sometimes call them, hydrogen NMR spectrum is the exact region that we see these exchangeable H1 of guanine and H3 of thymine. So when we see these, we know that they are in some sort of structure. There's a very indicative area that we can look at, for example, in values from maybe 12.5 to almost 14, you will get signals that are typical for a duplex. For signals that are below or to the right of 12 ppm, it's often the frequency we use, parts per million. In this region here, it's usually somewhat more non-canonical. It could be quadruplex, which is the example I show from here, but it can also be AG base pairs or something somewhat unique. So we have typical, Watson-Crick, and then something different. So already we can see from this simple experiment that we have not much folded character in the unbound aptamer. You can see there's not many signals because they're freely exchanging with the solvent. And when we add a ligand, we can see them very sharp. So there's folding. And we can also characterize it even from this few-minute experiment that there is probably some Watson-Crick and something else going on. So that was just a general example, but now I'm going to go into more detail for the actual aptamer we were studying for this particular project. As I mentioned, we took that very first DNA aptamer that was selected to bind dopamine. And here's another secondary structure prediction of the same DNA aptamer with a slight modification. If you remember the original one, if you can remember a few slides ago, there was a little bulge here. It now is just a full stem, but otherwise it's the same sequence. So if you take this DNA aptamer and do the exact same relatively rapid NMR experiment I just explained, we can see that in the unbound state, there is some structure, but it's the region that I already mentioned is typical of Watson-Crick. So we're basically seeing the stem of this DNA form, even in the absence of ligand, which makes sense because there are a lot of base pairs and you would expect it to form. However, there's quite a number of new signals or new hydrogens that appear when dopamine is added, including in the few maybe in the Watson-Crick area, but most of them in this more non-canonical area. So we know that, first of all, that the DNA does bind dopamine, but we highly suspected that already. More particularly, we know that it gets folded in some way when it binds the ligand. And even more, we know that it's maybe not a typical duplex formation. So it might already, even from this initial experiment, probably not look quite like the secondary structure prediction. Now, to do NMR spectroscopy, there's lots of peaks. In order to analyze these signals, you have to eventually figure out which signal is which atom. And the whole thing just becomes a lot easier if you can somewhat minimize what you're looking at. So the first thing we did was actually to eliminate a lot of that duplex region, because although it helped stabilize the fold in the absence of ligand, it didn't really add anything to our understanding of how the DNA bound to the ligand. So we tried many different deletions of base pairs in the stem, and we found that if you removed 7 of them, it's still completely bound to dopamine. So we still see the appearance of those dopamine-specific peaks, but it's much smaller. So you can see here that there are fewer signals, but it still has all the characteristics of binding to dopamine. So the next step is, as I briefly mentioned already, is we have to determine which hydrogens are these signals that we see so that we can start to get a more atomic understanding of what this DNA looks like and how it's binding to dopamine. Okay, so the best way to do that is, well, you could do some very long analysis of NMR spectrum, or like we do in our case, we actually synthesize a series of synthetic DNA in which one specific base has been isotopically enriched so that we can see it using different NMR spectrum. And typically what we do is we make the same DNA oligo, but with the isotope nitrogen-15 replacing the normal nitrogen, which is the isotope 14. By doing this, nitrogen-15 is actually very easy to detect using NMR spectroscopy. So if we only put a isotopically enriched guanine in one position on the oligo, and we do a particular experiment that lets us only see hydrogens that are attached to a 15N nitrogen, there's only one signal. And so we know that that signal right here is from guanine 9. So the numbering is based on the longer sequence. That's why it doesn't quite match up. So we can do the same thing by making another DNA. We now have this G labeled, and so we know that G13 is that signal. Again, you do it again, and you can see that the signal right beside G9 is actually from the H1 of G16. We keep going through all the different guanines, and we can assign the guanines that are in the one-dimensional spectrum, or we can even look at a 2D spectrum as well. But so now we know already some of the information, but we already see, as I mentioned before, is these here is the more or less Watson-Crick area, but this is not. So already we see there's something different going on. And we also have a guanine 36, which is far to the right of the spectrum. So that's just looking at the H1, but there's of course many more atoms we need to look at to gain insight into the atomic structure of the DNA. So actually, it's not as common, but if the amino group of guanine, so H21 and H22, are also involved in hydrogen bonds, they can sometimes be seen as well. It's not often seen for a typical duplex, but sometimes it is. So we were quite surprised that actually we saw many of these hydrogens in the particular DNA aptamer we were looking at. And so you can see it's in a different region of the spectrum. It's more, this value is 6 to 12, so it's to the right of these signals. But they're typical of amino groups. So that means there's a lot more hydrogen bond going on than there's usually expected in DNA structures. So we did the same sort of thing looking at thymine. So again, we were able to determine which of these signals in the NMR are from the different thymines. And we could do the same for the amino group on adenine. Again, you don't often see these hydrogens. So already still hinting at the fact that there's something very different going on with this structure. And finally, we can also see all 6 of the amino hydrogens from the 3 cytosine in the DNA. So this was great. This is a very good starting point because we know unambiguously what these signals are. And as I said before, it's already indicating that there's something different with this particular structure. And one thing that was interesting is during our synthesis, which sometimes if we've had these isotopic uh, phosphoramidates around for too long, they sometimes degrade a bit, they sometimes don't work as well. And so we had a few little problems with the synthesis. But what it meant is that we had a mixture in some cases where we had the full oligo labeled on T10, for example, and some that were shorter. And as it turns out, the ones that were shorter. So for example, if you try to label on T10 and it doesn't work, your oligo, the way you synthesize it, starts at the 3' end, and you synthesize it around like this, and then it will stop at T11 if it doesn't work for the labeling at T10. And usually these abortive trans phosphoramidite based oligos, they just don't bind to dopamine, so we can purify them away and there's nothing to see. But we were doing the screen very rapidly, and so we just took the unpurified DNA in this case, and very surprisingly, the DNA that was shorter by 3 bases, which really doesn't look like it should be able to make a sort of structure, actually bound very well to dopamine. So if you look at the top, this is the normal spectrum we see of the dopamine bound to the DNA of the dopamine aptamer. And in this case, you can see that there are new signals appearing, and this comes from this slightly shorter DNA. And if we went to the company, ordered all of our normal DNA again, but this time ordering the smaller oligo that just started at this thymine here, going to the thymine here. And actually, it looked beautiful. Again, there's fewer peaks because it's a smaller DNA, but it actually bound very well. You can now see that there really is no structure in the absence of dopamine. And what was, I think, most surprising is normally when we reduce the size of an oligo, we accept the fact that it will bind slightly weaker, but should still retain all of the atomic details of how it binds to dopamine. So when we started with the original dopamine aptamer and removed those 7 bases, it got weaker, but still bound dopamine. But now, when we made it even a little bit smaller, actually, it recovered a lot of that affinity again. So we had it smaller, which is great for NMR, and it bound tighter, which is also great. So this particular aptamer we called RKEC1. The initials are from the two PhD students in Philip's lab. And we used this for the structure determination. So it was a very nicely accessible size, and it really was a better affinity binding than the shorter one we were using before. So just to say that we can then look at a lot more information. I told you about the bases, but we can also look at sugars as well. So this is a two-dimensional NMR spectrum. It connects a hydrogen, and in this case, to a carbon. And these are the carbons of the sugar, for example, at the 3', 1', and 4'. And we assigned all these, which means that for the signal, we knew which atom it was in the DNA. And so we have a long list, and we knew all the different frequencies for all the different hydrogens in the DNA and for the dopamine as well. And then we did a type of experiment called a NOSI. And so if you've never heard of this before, it's an NMR technique we use to generate structures, because what it allows us to do is determine distances between pairs of hydrogen. And the closer the two hydrogens are, the more intense or larger the signal is. And if they're further away, it's less intense. And you can actually calibrate this and also give identities. So you'd know that, for example, this hydrogen is maybe on T12 and this hydrogen is G34. And you know that there are a certain range of distances apart. And so we do this for as many different cross peaks, we call them, these NMR signals that connect two different hydrogens. And we collect all of them and we make a list, and then we use a program. I typically use one that is perfect for biomolecular NMR structure determination. It's a combination of ARIA, which allows us to use some initially unassigned spectra in the initial stages, so it saves a bit of time and it has more freedom in the type of input we use. And then we use CNS, which is the program that actually does the structure calculation based on the information that we give it. And what's nice to see is this top number here. So the total NOE basically means the number of distance restraints we have between hydrogens in the DNA and dopamine that relate to some structural information. It's pairs of hydrogens that we know roughly the distance between them, and we can use that to determine basically the structure of the DNA bound to the dopamine. So what the program does is it minimizes the structure so that all of these distance restraints are satisfied in the final structures. And that's great. The only problem with this particular program, it's excellent for proteins. The force fields, or the way that it takes care of DNA and RNA, and in particular bases and base stacking, is not optimal. So we typically need to use another program afterwards. So this is when I start switching to the white background slides, because this is now using Amber. Um, and so what Eric now did, so that's why we were in the different background, took this ensemble of 15 structures calculated by the standard biomolecular NMR way, took them as well as, just go back for a second, as well as only these top distances that we knew unambiguously. So there's no dihedral angles and none of this ambiguous category. Just took those and used a restrained molecular dynamics. And if you have questions about this, this is why Eric's also connected. He can tell you some more details. But looking at literature, trying some things out, came up with the optimal model of the water to be used during the Amber molecular dynamics restrained simulations in order to get these final structures. Also, mimicking the solvent that we had, adding the correct amount of sodium and chloride, and then doing, for this part of the project, a relatively short dynamics run, mostly to allow the structure to relax a bit from the angles which may not be completely natural because of the other program we were using. So you can see here that they actually stabilize quite fast if you just look at RMSD based on the initial structures. And you can also see they don't change a lot from the initial structure as well. And after these 10 nanosecond trajectories, we took the final structures, and then we had a new ensemble of structures that were now much better properties in the structure, more relevant to how DNA actually looks like in solution. What this shows are how the different distance restraints distribute among the DNA and ligand. I'll show you the structure details in a second, but this is just to show that the large number of restraints really makes it possible to very accurately define how the structure comes together, even though we're only looking at hydrogens. So you can see that the distances are really focused at where the hydrogens are, but because there's so many of them, it's actually a very well-defined structure. And you can see that we have restraints both within the residue, so to help us to know how the conformation of that particular nucleotide looks like, between DNA bases and sugars, but also dopamine, the ligand within itself, and also between the dopamine and the DNA. So it's very well characterized, you can see here. And even though there's a lot of distance restraints, they're actually all satisfied, and there's none that have that are outside of the range that we give it, except a few cases where it's below 0.3 angstroms, which if you do structure calculations, is basically nothing. So everything is perfectly satisfied. So we're very happy with the structures we get. So there's 15 of them. You can superimpose them, and that's what we typically do. And we can now talk a little bit more about the structure itself. So here's a superimposed ensemble of 15 structures. You can see, most importantly, the dopamine located here in yellow. And maybe you start to see that there's possibly something a little more complex going on than a simple duplex and a bulge where the dopamine binds. This is relatively hard for me to explain from this point of view, so I'm going to do a more schematic version. And this will already maybe highlight that there's something more non-canonical going on, or at least it doesn't look like a duplex. So if we start at the T thymine 11, it goes towards the bottom here. Then you have a long stretch going up, passing uh, part of the dopamine interactions along the top where there's a loop that comes down. And I'll get back to all these details in a second. It goes back up. And then the dopamine is stacked between a thymine and a guanine base. And then you come to the bottom of the molecule. So you see that there's regions of 4 or 3 or 2 throughout the structure. It really looks highly different than the secondary structure prediction would have you believe. So it was really interesting and surprising. And that's why we're glad at the very start we did these very careful and unambiguous assignments of which signals came from which base because of all those different oligos that we made. So we knew that there was no error in the assigning of which hydrogens were close to other hydrogens. This was all completely unambiguous. So we're glad we did that because otherwise, you start to look at this and think, wow, that's a very complex structure, but it actually fits all of the NMR information that we had. So it's a very confident but unusual structure. And this unusual nature will become apparent even more right now because when we start to look into detail of how the different bases come together, we were, I think shocked is the right word, shocked that there was actually only 2 Watson-Crick base pairs in the whole structure. And so we were left with how to explain things. And I'll show you where the Watson-Crick are in a second. But what it became rapidly obvious to us is that we couldn't just say GC is here and there's a duplex region. We had to go to the RNA classification because RNA has been known to be more structurally diverse for a long time now. We know that there's in the ribosome, for example, you can sample lots of different base pairs going on in different sort of structures, and RNA itself is found in lots of different structural architectures. And because of that, we used the nomenclature that was used for RNA and its diversity for DNA. And I don't think this has been used before in this context, or at least it hasn't been needed to be used in this context, because normally DNA is a little bit more conventional. So when we do that, we're able to look a little bit more detail on what's going on. So as I said before, there's only 2 Watson-Crick base pairs. One of them is at the top here, which may help stabilize some of this top loop. But probably the most important Watson-Crick is shown here, which is also located here in the schematic. So this Watson-Crick base pair does 2 things. Probably most notably, it's GC, has 3 hydrogen bonds, it's relatively strong, and it seems to anchor the top of this loop so that it is in a fixed position. And it also served as a platform atop of which the aromatic part of dopamine stacks. So that's the Watson-Crick part, but it's obviously more complex. So if we look at it just on a few different levels going down, we can see there's the Watson-Crick here. There's a GG base interaction going on, and these pluses and minuses tell you whether the DNA strand is coming towards you, so plus, or away from you, minus into the screen. So a normal GC base pair has 1 strand coming towards you and 1 strand going away. These Gs have parallel DNA, so they both have pluses. And then we have here anti-parallel, so normal DNA, but it's a GG base pair. And just keep going down. We see a base triple, but this part here is a GC. It may look like a Watson-Crick, but because they're both plus signs, it's actually a parallel DNA. So this is a reverse Watson-Crick. And then interactions, hydrogen bond to this thymine 32. Going one step further down, we now see already some of the interactions with the dopamine. So this is through hydrogen bonding. And this is again an AT. Looks like a Watson-Crick, but because they're both in the same direction, it's parallel. So it's a reverse Watson-Crick. We then have a relatively almost normal looking part of the DNA architecture. We have the Watson-Crick that I've already mentioned here, but then there's a gap, and because of that gap, the bottom three bases, although they one of them is Watson-Crick-like, AT, but they're now parallel again. That's what these arrows show. So because they're parallel, even this AT is a reverse Watson-Crick. And then you also have GT and GG base pairs going on. And finally, you have a part that I think is actually quite interesting as well. It's very strongly hydrogen bound, but in a staggered way. So a lot of the interactions connect guanines more than other bases between guanine on the top and the bottom, using all of the possible, usually, well, different sides of the base, actually, Watson-Crick and the wobble side, to connect a kind of staggered architecture that links these back to the front. And the reason it's partly staggered is you notice here that there's four bases that kind of go alongside three on the other side. So it's not a perfect match, but it's very well hydrogen bound. And this already partly explains both the top and the bottom part, why we see so many exchangeable hydrogens that we don't normally see. It's because, well, they're not standard Watson-Crick. And in fact, all of them are involved in very strong hydrogen bonds. We then want to do a bit more information about the structures. So Eric first did some principal component analysis to see if we could take three diverse structures from the ensemble and do longer simulations to see how they behave. And that's what we show here. Whoops. So this shows now a microsecond simulation that the first, second, and third are using restraints, the same restraints we did use for the ensemble, and the fourth one is without any distance restraints, so unrestrained molecular dynamics. This is just a sampling of some of the structures, of course, over a microsecond. But if you then analyze it, then you can see something a little bit more useful and easy to interpret. So I'll get my pointer back again. And so if we look, for example, at the dihedral angles, it's an easy way to look at some properties during the microsecond simulation. We can see for these dihedrals, the beginning of the trajectory is in the center and the end is at the edge of the circle. And so you can see that they sample not too large variations in the chi-1 angles. So basically how the base is positioned with respect to the sugar. Usually they are all anti, but there is one guanine that is a syn, so it comes where the base comes on top of the sugar, and actually it follows the NMR spectrum, which has unusual chemical shifts or positions in the spectrum, so it all makes sense. It fits the NMR data as well. And what's interesting is these are from the restrained molecular dynamics. If you do the same thing without any distance restraints, we can see there's more variability, but the overall pattern is the same. So this is comforting. It shows that the restraints aren't causing any unusual limitation to how the DNA behaves. We can also look at other aspects as well. Just as one example, we can look at the sugar pucker. So to see how the different bases, how the sugars look like in the DNA. So these are during the entire microsecond. That's why it's a cloud of distances. The center is basically a flat sugar, and the edge is the highest amplitude of the particular dihedral angles of that sugar. So most of them are typical for DNA, C2' endo, but you can see that there's some other relatively unusual sugar puckers as well related to the fact that the DNA does a lot of complicated architecture. We can also look at hydrogen bonds, uh, again, just to see how stable the interactions are. And if you just look at these red or pink ones, these are hydrogen bonds between the DNA and the dopamine. So it shows you whether it's restrained or unrestrained. The binding mode appears to be relatively shared between the two and is consistent throughout the entire trajectory. So I'm going back now to the black slides because we're just going to look a little bit more at the dopamine itself. So the dopamine is bound here in the middle of the DNA. Because of the technique that is used, it's called capture selects. It's a way to select DNA for a ligand in which there is nothing, the DNA is the part that's attached at one point, but the ligand itself is completely natural. So there's no tethering. So a DNA that's selected using this technique can often bind completely around the ligand it's selected for. And that seems to be more or less the case here. So the dopamine is stacked on this GC base pair. And if you look here, it's then further, there's some important hydrogen bonds between thymine 18 and thymine 19 bases. And there are some interactions as well with the positively charged amine group, uh, with mostly the base and sugar of the G28 and A27, which is nearby. And if we look at it more to see an accessible surface, we can see that really only a small part of the dopamine is somewhat accessible to the solvent or to the water. A lot of it's quite buried within the DNA aptamer. And we can use this just to probe a little more the structure to make sure that it makes sense with the atomic details and the binding information. So we know that, for example, we can buy a analog of dopamine, which has a hydroxyl group in this position. And because it's completely external and pointing to the solvent, we don't expect to be much change in affinity. The initial affinity is 0.8 micromolar, and this is essentially the same. So that makes sense with the structure. In contrast, if you then start to put it in different positions, for example, if you put on the aliphatic part, the hydroxyl group, it's now a little bit closer to the surface. There's potentially some steric hindrance going on, and you can see a slight drop in affinity. This is much more dramatic for two reasons. When you add a methyl group deeper within the dopamine, so if you take the hydroxyl here and instead have a methoxy group, you can see, first of all, you're breaking an important hydrogen bond, so that clearly changes affinity, but you're also introducing more atoms in a position where there's really no extra space for atoms to go. So you have steric effects and a loss of a hydrogen bond, and the affinity is about 100 times less in this position. And then if you do the same thing but to the back, well, now you're affecting an oxygen that has two hydrogen bonds, and it's even more sterically hindered. So now it's almost 1,000-fold weaker in binding, but it completely makes sense with the structure. And we can also remove aspects. So if you take away the hydroxyl, it binds also 100 times weaker. And if you take away Yep, sorry. And then if you take away the aliphatic amine, then it just doesn't bind at all. So you may think that part's not important, but without that positive charge interacting with the sugar and base, but clearly also ionically with the phosphate backbone, it doesn't bind. And you can also shorten the aliphatic amine a bit, and it binds somewhat weaker. And maybe this is clear, but if you change the amine to our carboxylate group, so changing the charge, there's no binding at all. So all of this nicely makes sense. And we didn't want to just determine the atomic features of this DNA aptamer bound to the dopamine, but we also want to use it for something. So we collaborated with Philippe. This is the group in Sherbrooke in Quebec, Canada. And the typical way they make a biosensor from a DNA aptamer is to attach one end to an electrode, a gold electrode, and the other end to a something that can give you a change in electrical signal, in this case, methylene blue. And typically you have one on the 5' and one on the 3'. You can also invert it, but these are the two standard ways. And what we found is using this standard setup, there was really not much signal when you bound it to dopamine. Instead, if we looked at the structure that we had, we knew that this T, thymine 14, is really one of the few bases that's not involved in anything in the structure. And we knew that it would be the closest base also to the 3' end when you have the dopamine bound. So it's quite unusual to put the methylene blue inside the within the sequence of the biosensor, aptamer biosensor, but in this case, it worked much better. So it shows that already, even in the simple case, you could use the atomic information to better answer, how to make the DNA aptamer better. And finally, as actually Rachel already mentioned this was one of the targets in the CASP 16. Uh, One of the figures, maybe Rachel, you made this figure. I'm not sure. It's very nice. It shows color coded on how the structure should assemble, and you can see that for some different predictions, the similar colors aren't really coming together, highlighting a little bit that this particular DNA seemed hard to predict. And we have, of course, some reasons for why. Uh, One is that dopamine seems to be quite important for the actual folding of it, at least when you look at it in normal solution. But probably much more importantly, is the second fact here, is that most of the PDB, when it has DNA, has double-strand DNA in a duplex. You might have some quadruplexes or maybe something a little bit different, a stem loop as well, but that's it. So I think there's also a need for lots more structures of these um, aptamers in a way because they're selected and there's no reason to look a certain way. I think they will have a lot of diversity and they could really help come up with this different types of structures DNA can actually do. And so I'll leave this slide on the end. This is just highlighting that it's in bioRxiv. It hopefully is a paper soon, and you can already access the structure if you want.
All right, thank you very much for that very comprehensive and well explained presentation. I think NMAR is super hard to present, so thank you for kind of stepping us through it. For questions, feel free to raise your hands, guys, or put it in the chat. I'm gonna actually get us started on a prediction question.
Okay.
So we talk a lot about um, kind of data assisted prediction. And I think NMR comes up a lot in can we give kind of predictors the 1D spectrum, have them try to predict what the RNA is with that kind of data assisted prediction, and then use kind of 2D spectrum or more advanced NMR to kind of assess how well they did. What are your guys' thoughts on applying this to nucleic acid structures? Would
Yes.
it kind of be helpful in general for your experimental workflows? Yeah, any comments you have on that?
It's a very, very good question because ideally, exactly what you just said, I mean, a limitation is this obviously wasn't a rapid thing to do. And there are so many different DNA aptamers or other structures out there that if you could at least get a hint at the start, that would be great. I think it's quite solid, the Watson-Crick part that I mentioned in the 1D spectrum. So, I mean, naively, you could imagine just simply adding up hydrogens, so basically base pairs that are in the Watson-Crick part, and comparing it to even just a standard prediction just to see does it match the duplex region that you imagined? We thought a bit about this. I was working with a group that does um, detailed mutations of aptamers. And so they really just wanted a kind of a quick indication to know what they were starting with. And we tried that. It's hard to say. We're going to have to really go, I think, into a lot of examples to see how closely it goes. The second thing is we tried to maybe come up with a quick estimate for how folded a DNA is. So basically, maybe you could take an initial spectrum like I did. There'd be the way you do a lot of NMRs, you calculate the volume under the peak. It tells you how much hydrogens are there. And because these are exchangeable hydrogens, they can be 0 up to 100%. So it's not like the normal organic chemistry NMR where you can count up the hydrogens, and 3 hydrogens means you have a methyl group and things like that. So in this case, we wanted to see if you could quickly add the ligand and see the expected change of structuring, but it's been a little bit more complicated than it would appear. So, yes, great question. I think maybe, but we need work.
Yeah. All right. Next question, Nivedita. Do you want to read out your question? Here, I can read it out if you don't have. Okay, I will read out.
Oh, okay. I found it too.
So she asked, did you compare other water models for sampling with and without restraints for this DNA aptamer, like 3-point water models like OPC3 or TIP3P? I'm guessing this is a
Yeah,
question for Eric. Or you
exactly.
can
So he
answer.
just appeared now. So
Can
turn
you
on your
hear me?
microphone and go, Eric.
Yeah, can you hear me? Yeah, okay. So no, I didn't try tip 3P for this one. 4-point models are a bit more accurate in terms of water physical properties, and we have access to a very nice GPU cluster. So the cost of running a bit more complicated models is not an issue, really. Given that in these particular applications we have many NMR restraints, there's not some the DNA cannot do much actually. It's really well described at the experimental level. So I don't think the water model would impact the results significantly. And I'm seeing a second question actually.
Yes, second question
Yeah,
is also
on
relevant
GAF.
for you.
Yeah, I use indeed the GAF for dopamine, which is a very small molecule. So I didn't actually derive partial charges, and I didn't do a particular modified force field for dopamine. It was nicely covered with GAF.
I have a question that kind of reaches both maybe simulation and experimental. For RNA, we think a lot about kind of torsional space and specifically backbone torsional space. So thinking about pseudotorsions and how kind of the allowed backbone conformations restricts our kind of RNA conformations that are possible, but base pairs are possible, for example. What does DNA torsional space look like? Do we know much from experimental side? Is it mostly from simulations that we're able to determine it, and how does that kind of affect what's possible in DNA? Is it more flexible, I guess, when it comes to diversity of structures possible?
Yeah, that's it. It's interesting because going into this, I just really assumed that I knew that DNA was not flexible. Like I thought that was just a thing, and that's just how it was. RNA is more flexible. That's why it has all these great structures. I I'll let if Eric found a paper, I'll let him answer, but I couldn't find the the type of paper that I would like that would give a lot of this basically the what is the landscape that it can actually sample easily. I didn't really find this. Eric, did you find?
No, so I'm a big quadruplex guy actually. I would say by training.
Mhm.
um, so there's some diversity, but in a way, it's also contained to a very particular type of secondary structures. So there is some diversity in this, but I don't think people have explored so much what DNA can do. And I think that's because biologically relevant sequences tend to just behave very nicely and predominantly form duplexes. Even quadruplexes are not favored if the complementary strand is present. And I think, as Cameron said earlier, aptamers is really a good opportunity to explore what DNA can actually do in
Hmm.
terms of conformational landscape.
Yeah, and one of the um, I don't think it was for this one, but sometimes we, of course, try mutants. We change the base so that usually in a simplified way, you just don't want to bind the ligand anymore. So you gain insight on which bases are important for that. However, sometimes we notice that when you change a base, the effect is more that you get a different stable structure in the absence of ligand than changing necessarily the direct context to a ligand, for example. So I, th I think the fact that you can have all these different conformations argues a bit that it's more dynamic and more flexible than one would think.
Great. There's no further questions. We're just gonna thank Cameron and thank Eric for coming and talking about the super exciting structure. I hope we see more DNA structures coming up. Aptamers, I'm also, there's DNA viruses that could have it. There's lots of interesting things that hopefully in the
Yeah.
coming years we'll see
So,
more
so
of.
we are doing more structures. So hopefully the timing is such that you'll see one next time there's a CASP.
Yeah. Nice. The NMR is very hard, I'm sure.
Yeah.
So we'll see everyone again in 2 weeks, so 19th of March, I believe, for a talk by Pratush Tiwari. Thank you again, and the recording will be posted by the end of the week.
Thank you, Rachel. Thank you.